All right, welcome. So, as we begin to talk about evolution, we like to start with just talking about biology in general. So, this is a biology class, but it's focused more on the topic of evolution. So, just to get a background, we'll talk about some of the other things that maybe we won't really address throughout the semester as we're really focusing on evolution, but are important to understand in the discussion of evolution. So, biology is just the study of living things, the study of life. So, as much as chemistry is going to look at molecules and earth science, or geology are going to look at rocks and the earth, biology is talking about anything that is going to be living, anything that has life. So while the other sciences do play a role in living and life, biology is focused specifically on life. And of course, we're going to talk about the other ones because they're all intertwined, but we are talking about biology. When we're talking about evolution, it's a topic in biology. So living things are all descended from a single-celled ancestor. So when we talk about evolution, we're talking about a long period of time, gradual changes that have occurred over this long period of time. We're not saying that you descended from, or your great grandmother was an ape or anything like that. We're saying that minor changes over a long period of time have led to where we are now. So it all started very, very simple, and it became more and more complex over time. So the theory is that we have started from, or we started from single-celled organisms, and all organisms that are alive today are descendants from that single-celled organism. So the characteristics shared by all organisms lead to the conclusion that all life has a common ancestry. So all living things have similar, not similar structures, but they all have structures to them, and their functions have similar purposes to them as well. So some characteristics that we see in all living things: they're composed of common set of chemical components. So they're all using chemical components, and they all have similar structures. For example, cells. Also, more complex organisms are all going to have tissues, and even more complex organisms are all going to have organs. So we see this in all organisms. So as much as you might want to say that it doesn't seem like you could be related to a fish, if you look at just the general idea of the structure and the functions that are going on, it seems like we have a lot in common with them. So they also depend on interactions among complex parts that maintain life, contain genetic information. So they're all going to have DNA, and the fact that they all have DNA really points at this idea that we all came from the same origin. All right, they convert molecules obtained from their environment into new biological molecules, extract energy from the environment in order to use that energy. They replicate their genetic information in order to reproduce. They're going to share structural similarities among a fundamental set of genes, and they evolve through gradual changes in genetic information. So when we talk about gradual changes in evolution, people often say, hey, you know what, this doesn't really make sense to me. How could something go from being so simple to really becoming extremely, extremely complex, as we see in humans, or even if you see in a lizard or something like that, more complex organisms? How do you go from one to the other? Well, when we talk about this, we're really looking at 4 billion years. So the Earth formed about 4.6 billion years ago, we'll say. So 4.6 billion years ago, the Earth was formed, and then about 4 billion years ago, so 600 million years after that 4.6, you have 4 billion years ago is when life started. Now, life was not the way you picture life now. The Earth was not the way you picture Earth now. It was very different. Think of this desolate environment. All the habitats were very desolate. It was a huge void, really. In order for it to become what it is today, it took life to really reshape the entire Earth. So We're talking about 4 billion years of evolution to go from a single-celled organism all the way up to more complex organisms. So if we were to put this onto a calendar, put this into a month, we could say, hey, you know what? Earth was made on day 1 of the month, and then we see that life started on the 5th day of the month, and we didn't really see multicellular organisms, so everything up until the 24th day of the month. So you're thinking of this is billions and billions of years where it was just these single-celled organisms, very simple. But then eventually on the 24th day, we had these multicellular organisms. The multicellular organisms started to form tissues. Tissues could form organs, and then organs were going to form organ systems, and the organ systems were going to form these complex organisms. And then when we look at the last 4 days of the month, we see that you have this abundant fossils of aquatic life. So now the aquatic environment is becoming more complex. You have first land plants. First land animals aren't until the 28th day of that month. And we see that the first birds weren't until the 30th day of the month. And it wasn't until the last hours of that last day of the month that human-like organisms came along. Our first hominins or hominids came along. So in the big scope of things, mammals are a very young species, but more specifically, humans or humanoids or hominids, they are much. They're very young in the big scope of the Earth. We have not been here very long. Right? You're talking about 4 billion years. You're talking about the Earth is 4.6 billion years old, and we've only been here for hundreds of thousands of years. So complex biological molecules possibly arose from random associations of chemicals in the early environment. So we're talking about very simple organisms, very simple cells that they don't have all the organelles that you saw in your high school biology class, where we always see on the poster on the wall, it has all these different organelles. It has the nucleus, and it has the endoplasmic reticulum, and the mitochondria, and the cell wall. All these things are more complex cellular organisms. When we're talking about the first ones, we're talking about very simple organisms. We're talking about a membrane that is enclosing nucleic acids, enclosing like a DNA-like structure that has genetic information to it. So they've actually simulated this in a lab where over time, over a long period of time, if you have the right chemicals present, they're going to eventually come together and form nucleic acids, and they can form a membrane, and that's how the initial ones came together. And then after that, we see this slow progression of them being able to reproduce and create more complex organisms over a long period of time. We're not talking about 1 generation, 2 generations. We're talking about millions and millions and millions of years. So the next step was then to form a cell. So there were these nucleic acids, or you could think of it as DNA, and they formed a cell, and the cell was made from you had these fatty acids that created this membrane around it. You think of it as a cell membrane you might have learned in high school biology. So membranes were able to separate the cell, which was really just, think of it as that DNA or that nucleic acid, from the outside environment. So for 2 billion years, organisms were just these unicellular prokaryotes. All right, early prokaryotes. All right, these unicellular organisms that didn't really have these organelles to it. They didn't have a true nucleus to it, where all the genetic information was packed in tight into the nucleus. It was kind of just this membrane that contained genetic information. So for 2 billion years, we're talking about from 4 billion years ago to 2 billion years ago, there was just unicellular organisms. Think about it. Think of it as like a simple bacteria. That's what was on the Earth for 2 billion years. So our early beginnings, it took a very long time. When we talk about evolution, evolving from a single-celled organism, we're not, we're really saying that this took a very, very long time. So They were confined to oceans where they were protected from the UV light. The UV light was too damaging to these cells, so they had to be in the water. And there was no life on land at that point. So there was little or no oxygen in the atmosphere. So like I said, it's very desolate. No oxygen. Obviously, no plants because plants are living organisms. There were no plants. There were no complex organisms. Is really just this desolate place that had some land, had oceans, had water, and it had these little cells. Think of a little bacteria living in the water of the oceans. There was also no protective ozone because there was very little O2. There could be no O3 or ozone layer to form. So it was this very caustic, desolate area to really live in. Once photosynthesis came along, all right, those organisms, those single-celled organisms, were able to use photosynthesis. They were able to take in sunlight along with using the water, and they were able to make energy from that. And as a byproduct from taking in the CO2 in the environment and the water and the sunlight, as a byproduct, it made oxygen. All right, it's not something that they necessarily wanted to do, but that is what they did. So we have these single-celled organisms. You could think of them as cyanobacteria. Some people like to think of them kind of like as algae almost, and they were producing O2 not to help reshape the earth or anything like that, but that was just a byproduct. That was a waste product of the process of photosynthesis, and it started to accumulate as these organisms really thrived in this environment that had carbon dioxide, had sunlight, it had water. It allowed them to use photosynthesis in order for them to stay alive so that they could have energy. That was their source of energy. So with their source of energy, they were able to stay alive, and they pumped out this oxygen. So as it accumulated, solar radiation in the stratosphere broke O2, all right, or oxygen gas into ozone gas or O3. And it's a greenhouse gas that keeps the heat within the atmosphere. So when we talk about greenhouse gases, we're talking about ones that are going to maintain heat in the atmosphere. You hear a lot about carbon building up in our atmosphere, causing this greenhouse effect. Well, it is heating the environment. That's what's causing climate change. All right, it's a big issue. But once upon a time, when these organisms were around, it wasn't a big issue for humans then because humans weren't around, and it was good for creating this environment where there was oxygen available for the organisms that would come along that would use oxygen for their own way of getting energy. So O2 was actually poisonous to many of the early prokaryotes, and eventually you see that the organisms started to come along that were tolerant to the O2. So those ones that were tolerant to O2 or to oxygen, they evolved to use the oxygen in energy production, in cellular respiration or aerobic respiration or aerobic metabolism. They were able to use that, which was much more efficient than the anaerobic metabolism or anaerobic respiration. All right, the respiration that involves lack of oxygen. A lot more energy could be produced in aerobic respiration versus anaerobic respiration. So organisms were able to grow larger, and that aerobic respiration was able to provide the organisms with as much energy as they needed to carry out their life. So the ozone layer absorbs UV light, and its formation allowed organisms to move from the ocean to the land. Now, this is over billions of years, like I said. So think about, we're starting at a very desolate area where, or desolate habitat that is made up of water and land, no plants, no animals, no complex organisms. It's really all there is, and there's not even oxygen there. And over time, these photosynthetic organisms started to build up the oxygen and really change the dynamic of the earth, because now you have an ozone layer, so it's maintaining the heat. The heat is staying in. It's creating kind of a more stable environment to it. And some cells evolved membrane enclosed compartments called organelles. This is where we start to call them eukaryotes. So Previous to this, we had our simpler prokaryotes, and now we're talking about eukaryotes. So we are actually eukaryotes. The cells that we contain are going to have these organelles in them. They're going to have a nucleus, where prokaryotes don't have a nucleus. They don't have these organelles in them. They're very simple. So now we move on to eukaryotes, and eukaryotes are going to use mitochondria, and the eukaryotes that use photosynthesis are going to use chloroplasts, and this is a mode to create energy. So all living things, they need energy, they use energy, and in eukaryotes that is achieved through mitochondria and chloroplasts. So there's a theory that we talk that is that mitochondria and chloroplasts might have been simple organisms of their own, and the early cells encapsulated them in their cells and actually made it a part of them, and we call that endosymbiosis. So if you know anything about symbiosis, which is this idea that two organisms are going to live closely together, I always talk about Finding Nemo because most people have seen that movie. Finding Nemo, right? It's about a clownfish. Well, a clownfish, they have a symbiotic relationship with a sea anemone. The sea anemone is going to protect that clownfish, and the clownfish is going to provide food for that. So they are two organisms living very closely together. That's called symbiosis. Well, in endosymbiosis, we're seeing that an organism is going to encapsulate another organism, and they are going to live together, one inside of the other, opposed to the clownfish and the anemone, where they just live very, very close together. So we have this idea that mitochondria, chloroplasts, they might have been kind of these very simple cells of their own, or simple organisms of their own, and these early eukaryotes engulfed them into their cell membrane and said, hey, you know what, you're going to make energy for me, I'm going to keep you alive, you're going to keep me alive, and that's going to be our symbiotic relationship, and we call that endosymbiosis. So multicellular organisms arose about 1 billion years ago, and groups of eukaryotic cells probably failed to separate after division. So you have these eukaryotic cells that are sticking together, they're forming tissues, and when we start talking about tissues, we talk about cellular specialization, where cells start to become specialized to perform certain functions. So when we talk about cells, as much as you see in your high school biology classroom, you're going to have that model cell. Usually under it says animal cell, and they might have another one next to it that says plant cell. Well, that animal cell, it has a certain look to it on that poster, and it has certain organelles to it. But in reality, when we look at eukaryotic cells and we look at them in very complex organisms, multicellular organisms like us, or like a fish even, you're going to see that they're very different from one organ to the next, one tissue to the next, and that's cellular specialization. So evolution of species, how did all this change take place? Well, really there needs to be a set of directions how to build an organism. Well, how do we build an organism? Well, we need nucleic acids, and when we talk about nucleic acids, typically we're talking about DNA. So you need DNA, we need that list of instructions, but now that those instructions need to change over time, and that change in the DNA over time, we call that mutations. So some mutations, random errors in the instructions or in the DNA, those changes are going to give rise to structural functional changes in organisms, and those changes are going to eventually lead to new species. So as we talk about phylogeny, or the tree of life, the family tree of different organisms, you start to see that, okay, well, we had this one organism, and this organism, some of them started to have this trait, and some of them started to have this trait, and they kind of went separate directions as they started to live in different environments. And that's what we're really talking about when we're talking about evolution. Evolution is driven by natural selection. Natural selection is influenced by the environment. It's all the way that your DNA ultimately is interacting with the environment. How does your DNA interact with the environment? That's going to lead to evolution. So whenever we talk about different organisms, we like to use this binomial nomenclature, or this way of naming different organisms. As much as it's nice to say, oh, you know what, that's a dog. Well, you know what, when we say, hey, that's a dog, or that's a cat, that's a feline, that's a canine. Well, there's different types, right? When we talk about a canine, well, there's the common domesticated dog like you have as a pet, but then there's wolves, there's coyotes. If you're going to talk about a cat, well, are we talking about your feline, or are you talking about a domesticated cat, or are we talking about larger cats? So we use this binomial nomenclature in order to pinpoint its specific name. So as much as you might like to, call something, in around here we just say fox. If you see a fox around here, hey, but there's different types of foxes. So when you want to talk about what the specific type of fox is, we have this binomial nomenclature, and it's based on the idea of grouping organisms into these different sects, and it comes down to the most specific two is the genus. All right, so the genus is a group, and then the species is the specific one in that group. So Homo is a whole group of organisms, right? You have Neanderthals, were Homo neanderthalis. You have Homo erectus, and so Homo is the group. We belong to that Homo group, but we are sapiens. So it's always written this way, where it's in italics. The first word is capitalized. The second word is not, and that is how we always say it. So as much as we like to say humans, yes, we are humans, but Neanderthals were humans as well. Homo erectus, that was a human as well. So now when we want to talk about more specific things, we are going to use this binomial nomenclature in order to name them, to know exactly what you're talking about. So evolutionary relationships to species can be determined by comparing different genomes. And once upon a time, they like to create these phylogenetic trees, or these family trees, by just looking at how organisms looked, their morphology. But now we're able to look at their DNA, and we're able to really tell, hey, do they share these genes? Are they really related? So as much as they used to create these phylogenetic trees a different way, now it's really revolutionized as we can analyze DNA. So just as an example of a phylogenetic tree, it shows you, hey, early life started, and then it is going to branch out into bacteria and everything else. And then we're going to see another branch here. And eventually you get to these more complex organisms, and we end up here where we are animals. Okay, we'll talk more about this as we move forward, but just understand it's really a diagram to show you how organisms originated, which ones are more closely related than others. If we look at all of these organisms, well, they branched off more recently. All right, this area is more recently than this area, so we're more related to protists than we are to bacteria. So relationships in this tree of life are based on fossil evidence, structures, metabolic processes, behavior, and molecular analyses. Really, we'd like to look at molecular analyses. We like to look at the DNA. So there's three main domains of life where we talk about bacteria, archaea, and we talk about eukarya or eukaryotes. All organisms that are alive today descended from a common ancestor in the past. Living species did not evolve from other species living today. So we see that, hey, we came from common ancestors that are not alive today. So when we talk about the organization of life, we talk about very simple. You have molecules, right? Molecules are going to make up more complex molecules. Eventually you have these macromolecules, larger molecules, and then they are going to make up cells, and cells are going to make up tissues once we see cellular specialization occur, and tissues make up organs, organs make up organ systems, and then organ systems make up the organism. So organisms also interact with their physical environment and with each other, resulting in this hierarchy in a larger biological world. So in biology, we love to separate things into different categories, right? We like to talk about, hey, we're going to talk about evolution today. Tomorrow we're going to talk about genetics, right? We like to talk about, hey, this thing has organs, and when we look at an entire environment, there's different populations in there, but really all of them are interconnected, and they influence each other. So when we talk about larger hierarchies, we're talking about a species of individuals is going to make up a population, different populations make up a community. The community is going to live in a specific area, which is going to be known as the landscape, and all the different landscapes or all the different ecosystems are going to make up the Earth that we like to call the biosphere. So a system is a set of interacting parts or components. So the components are going to neither the parts nor the whole can be understood without understanding all their interactions or the processes. So in order to understand the process that's going on, or a process that's going on, or the processes that are going on, we really need to understand the individual components that are going on. So, if you were going to look at a specific component, we can't completely understand component A if we're just looking at component A. We also can't really completely understand component A by just looking at its interactions with component B, because component B is influenced by component C. So we really need to look at all of them in order to understand the entire system. And that's what biology tries to do. It tries to understand the entire system. As much as it's important to understand the physiology of a single cell, well, there's so many things that influence that single cell. You really need to understand what's going on outside in order to understand what's going on inside. So when we look at it on a cellular level, and we're going to look at the amount of free amino acids, right, the building blocks of proteins in a cell. Well, they're going to synthesize proteins, and the proteins are going to break down, and the amount of breakdown of products in the cell is something that we can look at. So by looking at these different components, we can really understand what's going on with the amino acids and the proteins in a cell. The same thing goes for if you look at an entire organism. Well, entire organisms, they have different organs, and the different organs are going to influence each other. So the amount of sodium in the gut from foods, the absorption of sodium from the gut by the gut cells is going to occur. The amount of sodium in the body fluids is going to change. That's going to lead to the kidneys having to excrete some of the sodium, and then the amount of sodium leaving the body is going to increase as well. So we see this effect where, hey, how do we regulate the amount of sodium? Well, people might say, oh, we regulate it, we make sure that we take in enough sodium. But there's so much more to it, because if we take in too much sodium, then we need to excrete it as well. So there's more to the system than just one component. You have to look at all the components to understand the entire process. And then on the community level or the ecosystem level, you're going to look at grass, and grass is going to be influenced by the voles or the mice in the area, and they are going to be influenced by the predators or by the consumers, whether it be foxes or owls. So this is how we like to look at a system in biology. So they're very dynamic. All right, rapid flows of matter and energy are occurring. They constantly exchange energy and matter with their surroundings. But there is kind of some persistence to it. There is rhyme or reason, and we can use it as a way to predict the future and understand what occurs in an environment. So when we talk about evolution, you really want to keep going back to this idea that it is heavily influenced by genetics. Now, this is not a genetics course. We're not going to dive completely into genetics. Genetics is a whole nother semester you could take in order to look specifically at that. There's so much to know about genetics. But when in our discussion of evolution, you do want to understand that genetics is really the foundation of evolution, because as much as we're looking at the big picture where we're seeing the traits and how the traits are interacting with the environment, well, those traits come from the genetic instructions. They're coming from the DNA, which is saying, hey, how to build this individual. So DNA consists of repeating subunits called these nucleotides. The nucleotides are like the letters, so you always learn about A goes with T, and T goes with A, and C goes with G, and G goes with C. Well, those are your nucleotides, and a gene is a set of those nucleotides. So we can look at, say, this set right here and say, hey, that's a gene that codes for something. So it's a specific gene is a specific segment of DNA that contains information for making a protein, right? And proteins make us up. So these these genes are going to say which amino acids should come together. Amino acids form proteins. Proteins are going to form cells. Cells are going to form tissues. Tissues form organs, organisms, organ systems, and then organisms. So proteins are going to govern the chemical reactions in cells and form much of an organism's structure. So as much as DNA is going to build these proteins, cells are the building blocks of organisms. So here we have our nucleotides. So one nucleotide right here, and nucleotides are going to make up this DNA. This DNA is this double helix, this twisted ladder, and the twisted ladder is going to have segments that we call genes because they are going to build specific proteins. And all cells in multicellular organisms contain the same genome, but different cells have different functions. So every cell in your body that has DNA is going to have the same DNA, but each cell uses different sections of that DNA. Someone once explained it to me. It's like if you think of it like a book, and you're going to have say white blood cells are going to use one chapter of the book to know how to be a white blood cell, and you're going to have liver cells are going to use another chapter of the book. So that every cell has every chapter to that book, but it's only using the chapter that it needs. So mutations are going to be the changes or the alterations of the nucleotide sequence, and when you change that sequence, that's changing the instructions, and that's going to change what proteins are going to be made. And it could be it could have a harmful effect, it could have a helpful effect, it could have no effect at all. But mutations are really what is changing the genes, changing the traits, and those traits are then interacting with the environment, and that's what's causing evolution or changes to the organisms over a long period of time. So evolution is just the change in the genetic makeup of population. When we talk about we say that individuals don't evolve, populations evolve. So if just one individual has a mutation, that doesn't mean evolution took place. That just means, hey, that organism had a mutation. But if suddenly that organism has a new trait, and that new trait is beneficial to them, and it helps them to survive and pass on their traits, well, that's going to allow them to live longer. That's going to pass on that mutation. And over a long period of time, you could start to see that population over many generations might start to have that trait more and more often, and that's where we eventually say, hey, the population has evolved. An individual doesn't evolve, an entire population evolves. So a common set of evolutionary mechanisms applies to all organisms. This idea of things changing gradually over time and their genetic makeup changing, that applies to all organisms when we talk about evolution. There are other things in evolution where it might be specific to certain organisms, but when we talk about just the basic principles of evolution, it applies to all organisms. So the constant change that occurs among the population gives rise to all the diversity of life. In science, we love to look at inquiry or doing science in order to understand the world around us. So observation nowadays, right, once upon a time, very simple. Now it's really enhanced by technology. We use microscopes, genome sequencing, imaging, satellites, all these different things. Now we're able to analyze DNA, something that once upon a time you never could have done. You could barely look at DNA and you weren't able to sequence an entire genome. Now we have technology that allows us to do this. So when we do our observations, it must be quantified, right? We need to be able to take in data, and we have to be able to measure. It has to be measurable, and mathematical and statistical calculations can be applied to it. So I'm sure you know from high school, you learned about the scientific method. Excuse me. And the scientific method is very simple in high school. In reality, it's not as simple as that, but for our purposes, we can still kind of think of it as a simple process where, hey, we're going to make an observation, we're going to then ask a question, and then based on that question, we're going to come up with a hypothesis or a statement. I like to say it's a statement because sometimes people like to phrase their hypothesis as a question. A question is not a hypothesis. A hypothesis is a statement saying this will occur. Whether it's right or wrong, you're making a statement. You're not asking a question or anything like that. And then you're going to have predictions and testing along with that. So once we come up with a hypothesis, we are going to then be able to conduct experiments, and we're going to use that quantifiable data to test our prediction and come up with a conclusion based off that. Now, this is how we learn it in high school, and it's very simple, but there's more to it because you know what? Our conclusion isn't always what we thought it was, and every experiment's going to, or any good experiment's going to lead to more questions. So you never end an experiment thinking, all right, I'm done, I solved my problem. Any biologist, any biology professor you're ever going to have that has a PhD that's working on something, whenever they finish something, they're going to publish it. Once they're done publishing it, usually they start right back at square one and they want to figure something else out because their last experiments led to even more questions. So what happens is, if you have significant results supported by a hypothesis, then you're going to repeat it, and and other researchers should be able to repeat it as well, further prove your point, and then you're going to ask other questions. If there, so you're going to ask new questions. If it does not support your hypothesis, then you have to re-examine it. You're going to revise your hypothesis, and you're going to start back going down that way. So it is more complex than maybe you learned about it in high school. So to answer questions, you're going to look at hypotheses, and typically we're going to have a null hypothesis, and you are going to have an alternate hypothesis. And your null hypothesis is just going to say this will not have an effect. Your alternate hypothesis is going to say this will have an effect. So when we look at our experiment, we can always have a null hypothesis and an alternate hypothesis. So we like to do controlled experiments because we can sit in a lab and we can make sure that all the variables are the same except one. Whenever you do an experiment, you only want to change one variable. If I were to test a new drug, okay, if I came out with a new drug, I wouldn't want to give a new drug to this group and also give them some old drug with it, and then give the other group neither drug. Well, you wouldn't want to do that because then I don't know, is it the old drug and the new drug, or what is it? You want to be able to just look at one variable. So when you're testing drugs, you want both groups, your control group and your experimental group. Remember, your control group doesn't get that drug. They're going to get a placebo. So you want everything to be the same in that control group except that one variable. And in your experimental group, everything the same except that one variable. So your control group isn't going to get that variable, and your experimental group is going to get that variable. So if you are going to do a test on plants and you want to see if this new fertilizer is going to work, well, you're going to say, hey, this group's not going to get the fertilizer, but they're going to get everything else that the other group gets, and this other group does get the fertilizer. You don't want to say, hey, this group's going to get this new fertilizer, a different amount of water, a different amount of sunlight. You don't want to change all these variables. You just want to change one variable. So for an example, you look at, say we come up with the hypothesis we want to study atrazine. So we have this chemical atrazine, and our hypothesis is that exposure to atrazine during larval development causes abnormalities in the reproductive tissue of male frogs. So this would be our alternate hypothesis. And if we were to do our null, so this would be our alt. If we were to do our null, we would say no effect. All right, so we would say exposure to atrazine during larval development does not cause abnormalities. That would be our null. So we're looking at our alternate hypothesis, and we need to have a list of steps, our methods, and our procedures that's going to occur so that once we're done with our experiment, we can tell another scientist, hey, look, this happens this way. You could do it yourself. Repeat the study. It'll happen that same way every time. So we have a list of instructions. And in our procedures, we say that we're going to have 9 tanks in which all attributes are held constant except the water's atrazine concentration. We're only changing one variable, and that's going to be the atrazine. You don't want to change the amount of water in one, the amount of salt in the other, the size of the tank, nothing. Everything's the same except that one variable, which is our atrazine concentration. So we established 3 atrazine conditions, 3 replicate tanks per condition. One has 0 parts per billion, which is going to be our control, the one that doesn't get the atrazine chemical in there. Then you have 0.1 parts per billion, and then you have 25 parts per billion. And we're going to put Ronopippians, tadpoles, that's a type of frog. Remember, if you look at how it's written, this is the specific binomial nomenclature for it. So those frog tadpoles from the laboratory, which are reared eggs in the 9 tanks, we're going to keep the amount of tadpoles the same. All right, so that's another variable we're keeping the same. You don't want to put 10 in this one, 20 in this one, 40 in this one. You want all of them to have the same amount so that when we look at how many survived, we have something to really be able to compare to. So when tadpoles have transitioned into adults, sacrifice the animals and evaluate their reproductive tissue. So we kill off the frogs once the tadpoles become frogs, and then we're going to look at the reproductive tissues and we're going to record our data. So we're going to test for relationships between degree of the atrazine exposure. Remember, we have 3 degrees of exposure, no exposure, 0.1, and 25. And we're going to compare that to the presence of abnormalities in the gonads or the testes of the male frogs. And their conclusion was exposure to atrazine at concentrations as low as 0.1 parts per billion causes abnormalities, all right, shown here. Look, if you're looking at abnormalities, none, right? If you're looking at abnormalities, now it's going up. It's going up. So you're looking at these abnormalities, it's increasing as time goes up. The effect is not proportionate to the level of exposure. So you would think 25 would be the highest. That's just not the case. But they do see that, hey, without atrazine, we didn't record anything. But when we do have atrazine, we recorded abnormalities. So when we look at doing an experiment, we're always going to have our independent variable, the one that we know, the one that we're able to manipulate. So before I do the study, I know that, hey, I'm going to have this concentration, this concentration, this concentration. Before I record any data, I know that, hey, my data is going to be looking at 0 parts per billion, 0.1 parts per billion, and 25 parts per billion. What I don't know is the amount of abnormalities. That's going to be the dependent variable. And whenever I'm going to plot it, this is going to be on my y-axis, and this is going to be on my x-axis. So if I go back for a second, you see that our independent variable is down here, right? I knew that before I started, and my dependent variable is going to be right here. I didn't know the amount of abnormalities. So x-axis, y-axis. Now, not all experiments can be done in a lab and are perfectly done and removed from the outside environment. So we often have to do comparative experiments where we look at differences between samples or groups. So the variables cannot be, like we say, controlled. We like to do these controlled experiments. Sometimes we have to do comparative experiments, and data are gathered from the different groups, and they're going to be compared. So if we say that, hey, the presence of atrazine in the environmental water correlates with abnormalities in the frog population, then we would set things up a little bit differently because now we're talking about environmental water opposed to setting up water in the lab. So based on commercial sales of atrazine, we found there are 4 sites that are polluted with atrazine, or we found 8 sites, and you have ones that are less likely and 4 sites that are more likely to be contaminated with atrazine. So we visit all the sites in the spring to look at the tadpoles, and we collect frogs and water samples. And in the laboratory, we kill the frogs, from the different sites, and we look at their reproductive tissues, and we document any abnormalities. And then we're going to analyze the water in order to make the correlation or the connection between the abnormalities that we see and what type of water or the amount of contamination that they were living in. And then we are going to analyze our data. So their conclusion was that the reproductive abnormalities exist in frogs from environments in which the atrazine concentration is 0.2 part per billion or above. So anywhere where they took these tadpoles or these frogs and they looked at their reproductive organs, they any ones that had abnormalities, they then were able to look at the water samples and see, hey, you know what, those came from the waters that had atrazine in them. So statistical methods help scientists determine if differences between groups are significant. Statistical tests start with a null hypothesis that no differences exist, and statistical methods eliminate this possibility that results are due to random variation. So as we move forward, keep these things in mind, because as we're talking about science, which you're in a biology class, evolution, we're talking about science. How is science conducted? How do we test our hypotheses in order to come up with a conclusion? All right, so as we move forward, you want to think about these things. Not all forms of inquiry into nature are scientific. Scientific hypotheses must be testable. We always want them to be testable. And have the potential of being rejected. So it's okay if your hypothesis was wrong, you have to reject it, but that is part of doing science. So science depends on the evidence that comes from reproducible and quantifiable observations. And I always talk about, if you go on Facebook, you see that there's a lot of people talking about alternative ways of doing things, and I always say, they're like these hippie moms that say, oh, I don't believe in antibiotics, I want to use essential oils, which if you believe that, that's fine, but there's just not any scientific data to back that up. So it's hard for people to get behind it, or hard for science to get behind it, because there is no data to back it up. So as much as you might feel like that is helping your child more than the antibiotics, there's no science to back that up. So until you can prove that, you might sound a little silly. So religious or spiritual explanations of natural phenomena are not testable and therefore are not science. Science doesn't say that religious beliefs are necessarily wrong. I'm not going to tell you, if you don't believe in evolution, I'm never going to fight you on that. But this course is about evolution, and we're looking at the science behind it and the proof behind it. So you don't have to necessarily believe it, but you have to understand that in the world of biology, this is what the facts are. And if you are going to try to fight that with your religious views, there's really no way to debate each other, because religion is based on beliefs, opposed to science is based on tangible facts that are going on. So keep these things in mind as we move forward and we continue to talk